Hello, and welcome to today's webinar entitled Accelerating Cell Line Development High-Throughput FC Fusion Screening with Cyto-Mine Chroma. I'm Colette Cooley, and I'm delighted to be hosting today's session on behalf of Sphere Bio. At Sphere Bio, we're committed to driving innovation in single-cell analysis and biopharmaceutical research. And today's webinar is an exciting opportunity to share some key insights with you. But first, some housekeeping notes. Today's presentation will last approximately 45 minutes and will be followed by a Q&A session. If you have any questions during the presentation, feel free to submit them via the chat box and we'll address them at the end. The recording of this session will also be available afterwards, so don't worry if you miss anything. Now it's my pleasure to introduce our speaker, Min Mo, one of our senior field application scientists from the US. Min, over to you.
Hello, everybody. My name is Min. I am a senior field application scientist with Sphere Bio. Thank you very much for attending today's webinar. So today I am presenting accelerating cell line development, high-throughput FC fusion screening with Cyto-Mine Chroma. So who are we? So Sphere Bio is a company based in Cambridge, UK, with fully staffed EMEA, US, and Asia teams. So our mission since our founding has always been to find the cells you want with ease and speed. So currently we have over 300 customers globally with a big IP portfolio consisting of 153 patents, and the list is growing. So we are a company with innovation at heart. Now, during this talk, I will first briefly touch on FC fusion proteins and the challenges that we are facing in finding the cell lines that express high amount of FC fusion protein. Then I will show you how Sphere Bio's pickle droplet technology can be used to tackle these challenges and find the cells you want fast using our Cyto-Mine system. Last but not least, I will showcase the Cyto-Cellect fusion protein detection kit. So here is a typical antibody composed of the Fc portion and the Fab portion. So Fc portion bind the cellular receptor on the cell surface, whereas the Fab portion bind the antigen. This is a typical Fc fusion protein, which is basically a recombinant protein consisted of Fc portion of an antibody molecule fused to another protein or peptide or receptor of interest. So there are a lot of added benefit of doing so, just to name a couple, right? So it increased the half-life of the drug and also increased the stability, solubility, and effective function of the fusion part. Good example of FDA-approved Fc fusion proteins is the etanercept, which is used to treat autoimmune diseases such as rheumatoid arthritis. So the Fc portion of this fusion protein is from human IgG1, and the fusion part is the tumor necrosis factor receptor 2. So the presence of Fc portion extend the half-life of the drug in the bloodstream from just a couple hours to 4 to 5 days, thereby enhancing its therapeutic effect. Due to the popularity of the Fc fusion proteins, there have been a big demand for Fc fusion protein. So in 2023 alone, FDA has approved 13 fusion proteins with roughly 40 clinical studies. However, the production of Fc fusion protein can be challenging and difficult. For example, compared to a standard monoclonal antibody, the expression and the yield of Fc fusion protein can be variable. Therefore, it is critical to isolate and find a stable cell line that express high amount of Fc fusion protein for downstream production purpose. But unlike monoclonal antibody, with which screening can be standardized using a Fab pair that bind the Fab and Fc portion of the antibody respectively, there is a lacking of standardized universal screening method for Fc fusion protein, which make the screening of the cell line expressing high amount of the Fc fusion protein very, very difficult and challenging. In addition, the standard workflow for cell line development, CLD, is not making finding these cells you want any easier. So here is a cartoon representation of the typical workflow for CLD work. So in order to isolate cells, for example, CHO cells or hybridoma lines that express high amounts of Fc fusion protein from a huge heterogeneous cell population, you have to follow these steps. First, single cell isolation, then functional screening, then selection and recovery. So these steps in between can take anywhere between 2 to 3 months to complete. So it is very tedious, labor-intensive, and costly work. So how do we find the best cell line express high amount of Fc fusion proteins fast? So at Sphere Bio, we make it easy because our mission is to find the cells that you want fast with ease since the founding of our company. So we have a lot of experiences in doing so. So using Sphere Bio's Cyto-Mine system, which I'm going to talk about very, very soon, you can isolate the cells you want in just 1 day or even a couple hours with ease and speed. So here, shown here is a Cyto-Mine Chroma. So Cyto-Mine Chroma is a second generation Cyto-Mine. So not only the Chroma retain the ease of use speed of the first generation, but also contains 4 lasers, which are shown here, and the 4 detectors, allowing true multiplex capability, so that you can isolate the cells you want in a more granular fashion. Under the hood, Cyto-Mine uses pico-droplet-based technology. Here is a cartoon representation of a droplet within which a cell and its growth media. So it is about 100 microns in diameter, approximately 450 picoliter of volume. So to put it in perspective, a CHO cell is about 15 micron in diameter. So our droplet is very, 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 very tiny. So below is a video showing pico droplet generation in real time. So cells and media are injected horizontally and pinched off by oil injected from above and below, resulting the formation of cells and its growth media. Here, this table showcases the feature and the benefit of our droplet. For example, our droplets are gas permeable to maintain cell viability, so the cells can grow, live longer in our droplet. Cells are encapsulated so that the secreted product, for example, antibody or fusion protein, can be measured. So this cannot be done using a standard flow machine. Size of the droplet is only 450 picoliter, which allows for rapid detection of signal for encapsulated cells. So this is a very important point, which I'm going to touch on again in the next couple of slides. And the droplet is clear and is compatible with fluorescent-based cell functional assay. Last but not least, our droplets are very, very stable. So there's a minimum fusion or breakdown during the machine time. How does Cyto-Mine Chroma work? So this is the typical workflow of Cyto-Mine to find the cells that you want. So process start with a population of cells in its nutrient-rich media, typically up to 400,000 for best single-cell output. And then those cells are loaded into a sample loader. Cells are then encapsulated in pico droplet surrounded by oil. So next, the droplets are pushed into an incubator and incubated at 37 degrees for up to 24 hours to allow biological processes to take place, such as fusion protein production or antibody production. Cells are then cooled post-incubation to stop further expression. So next step is sorting. So where molecule of interest are fluorescent labeled. So high producing cells give a high level of fluorescence, and we can tell the chroma to sort those cells only. Shown here is a typical thread analysis being used to find the CHO cell expressing high amount of antibody. So the FREP here, as shown here, bind the Fc portion and the Fab portion of the antibody respectively, and the energy transfer occur. The gated region on the left-hand side on the scatter plot contain the droplet with cells that express antibody. which are then sorted. So next step is dispense, which means that you can gate on the sorted cell population again to fine-tune the cells that you want prior to dispense. So with Chroma, you can now measure up to 4 fluorescent signals, right? So that to find the cells you want in a more granular fashion. Now, cells are then dispensed into the microtiter plates with each cell's image 5 times, as shown here, to ensure and demonstrate monoclonality. So to fill up a 96-well plate, it only takes a couple minutes, which unlike other platforms out on the market, which take much longer. So the heart of the technology is the cartridge. So where all procedure I talked about in the previous slides take place. So the cartridge is a single usage consumable. There's 2 major advantages, right? So first, reduce the chance of cross-contamination. Second, it increases system stability because all the complex fluidic take place on this single cartridge. So you use it and then you toss it, and saving trouble down the line. So here is the product portfolio currently offered by Sphere Bio to help you find the cells you want. It includes the Cyto-Mine Chroma system, consumable and reagent for your need in, for example, antibody discovery and the CLD work. Our software not only allow you to analyze data, but also offer traceability for every single cell that you have isolated for compliance purpose. The Chroma is perfect for CLD work. It increases speed. So you can load up to 400,000 cells per run for best single cell encapsulation. This compresses your timeline by weeks or even months to just 1 day or just a couple hours. And the system is very easy to use, very robust. So there's no need to worry about the fluidic mechanic issues, which allow you to focus on your biology, right? And it also reduces costs. Our pico droplet is only 450 picoliter per volume. So it is not that much, even if you screen millions of droplets. So saving reagent cost. In addition, reduced time in CLD workflow means reduced costs. Last but not least, it makes cell screening routine. So Chroma can interrogate large population of cells at high speed at single cell level with monoclonality assurance. So this combination is just impossible with methods such as manual dilution. So now you have a good understanding of our technology. Let's talk about the Cyto-Cellect FC Fusion detection kit developed by us to be used in conjunction with the Cyto-Mine systems. So the kit consists of a preformed FRET acceptor and the FRET donor pair, as shown here. So the FRET donor is a human FC labeled with Alexa fluorescent 488, which is green. And the acceptor is a FC binding protein labeled with Alexa fluorescent 594, which is red. So in the presence of FC fusion protein, the interaction of this preformed pair of FRET probe is disrupted, resulting in decreased FRET signal, which can then measured by our Cyto-Mine system. So this assay is a competition assay. To your left is a cartoon representation of what is happening inside a droplet, right? The cells producing fusion proteins in the presence of FC fusion detection probe. The pico droplet basically turns into a miniaturized bioreactor, drastically accelerate the buildup of FC fusion protein. So within just 1 or 2 hours of incubation, reduction of FRET signal can be observed. And this is because our pico droplet is only 450 picoliter of volume, which I mentioned in the previous slide. We can functionally screen millions of these bioreactors in a very, very short period of time, within just a couple hours to find the cells you want. So it would have taken a long time for cells to secrete enough biomolecule of interest to be detected in a traditional microwell format. So now let's look at the data. So just leave this cartoon here to remind you that our FC Fusion kit is a competition assay. So as part of our validation procedure prior to Cyto-Mine assay, we did a plate reader assay. So commercially available FC Fusion proteins, as shown here, were titrated in the presence of Cyto-Cellect FC Fusion probes. So abatacept is a commercially available FC Fusion protein, which has been used to treat autoimmune diseases, just like the etanercept. So the abatacept FC silent mutation is a very important FC fusion variant designed to avoid immune system activation, so making it potentially safer for therapeutic use in conditions where immune activation is undesirable. So this is a very, very important variant of an FC fusion protein. So as you can see, the FRET response was normalized to the response of 0 nanomolar FC fusion protein control. So the y-axis showing the normalized FRET response calculated by red intensity divided by the green intensity, whereas the x-axis shows the concentration of FC fusion protein in the assay. As you can see, increasing concentration of abatacept and etanercept result in decrease of FRET response, consistent with these molecules displacing the FRET donor from an acceptor probe, as shown in this cartoon, right? So importantly, the FC silent mutation in abatacept has the same potency in disrupting FRET as its wild-type counterpart, which means our kit can be used to screen for FC fusion protein variants, such as the FC silence variant. Figure B shows the FRET response observed over time, right, from 30 minutes to 180 minutes. A short incubation of 30 minutes, which is this blue line here, was sufficient to induce a significant change in FRET signal compared to the highest concentration of abatacept tested. So this kit works in plate assay format, and now we can move to the next step. So now we want to show that FC fusion detection kit works in our pickle droplet-based technology. So this is the assay setup. Known concentration of abatacept was co-encapsulated with our fusion FRET probes. And here is the scatter plot of the results. On the x-axis is the green donor. On the y-axis is the red acceptor. As you can see, all 6 pickle droplet population generated can be distinguished with a decreased FRET response at increased abatacept concentration from 0 nanomolar to 150 nanomolar. Right? As you can see, abatacept induced a significant drop in FRET signal at concentration as little as 9.4 nanomolar. So this assay is sensitive. So this data confirm that a FRET competition assay can be successfully applied in our pickle droplet technology. Now let's look at the Cyto-Mine results. Right? The FC fusion kit was used to detect and select FC fusion protein secreting CHO cells, mimicking a real-world scenario. So on the x-axis is the donor green signal, and the y-axis is the acceptor red signal. The CHO cells were incubated for either 1 hour or 2 hours at 37 degrees for fusion protein to be produced. So every single dot you're seeing here is a droplet, it's not cell. So this population here on the top left is the population of droplet containing the intact FRET pair. The presence of the cells expressing FC fusion protein is indicated by a drop in the acceptor signal, which is on the y-axis, and the increase in the green signal, which is on the x-axis. Those populations are gated and then sorted. So the longer the incubation, the more FRET signal being disrupted, as seen in the 2-hour incubation compared to the 1-hour incubation. The sorted droplets were dispensed in microtiter plates at 1 second per droplet, so that a 96-well plate, it only takes a couple minutes to complete. So next, we want to see if these single cells being dispensed can grow into colonies and produce Fc fusion proteins. So cell outgrowth was monitored for up to 2 weeks. About 54% of the cells dispensed remain viable and grow into colonies. And subsequent screening of more than 600 colonies show 89.8%, basically 90% of the clone, secrete the desirable Fc fusion protein. So I want to quickly emphasize here again, right? So the screening and isolation of the cells only take a couple hours to complete using Cyto-Mine system. In this case, had we not using our Cyto-Mine, it would have taken weeks or even months to find the cells you want, which is tedious and costly work. Now, the commercially available Fc fusion protein, which I mentioned in the previous slide, contain a human IgG1 Fc domain, right? So therefore, we want to see if our kits can detect Fc fusion with different IgG class Fc domains. So we test human IgG 1, 2, 3, and 4 antibodies in a plate reader assay. So I don't have time here to show you the results, but they are available in our app notes on our website. So in a nutshell, our kit can detect human IgG 1, 2, 3, and 4 in a plate-based assay, indicating our kit can detect Fc fusions with different IgG class Fc domains. So this result, the data shown here, is for a Cyto-Mine run. This data shows the threat response of CHO cells secreting human IgG4 kappa following either 1 hour or 2 hour incubation of cells with our fusion protein detection kit using Cyto-Mine. So Fc Fusion Pro were able to detect human IgG4 kappa secreting cells. Again, right, the gated region here are the droplets containing cells secreting IgG4 kappa. And of course, the longer the incubation, the more signal can be detected, right? So this together with the plate reader assay, which I don't have time to show, demonstrates that Fc Fusion kit is able to detect Fc fusion with different IgG class Fc domains. All right, so here is just a quick summary slide showing the key advantage for functional cell screening using our Cyto-Mine system. You can screen up to 400,000 cells for a best single cell output, or 40 million cells for pooled output, so that you can find the cells you want in just a couple hours. You can sort and dispense based on functional fluorescent assay of secreted product or surface marker. For example, if you want to screen B cells in antibody discovery workflow. So with Chroma, now you can interrogate cells with 4 lasers simultaneously. Cells are viable. Right? Cells are viable in our droplet for maximum outgrowth because our droplet is gas permeable. Now, our software has built-in monoclonality assurance. So 5 images are taken for every single cell dispensed. Last but not least, so this system is fully automated and then compress months of work into a single day or just a couple hours, or saving time and costs. The only hand-on time is preparing cells, and once they are loaded, it is taken care by our machine, and all you have to do is to set up the gate for dispense and sorting, and it is that simple. Now, before I end, I just want to quickly outline the kit that we have to help you with finding the cells you want. So we have the Cyto-Select Fc Fusion, which I just talked about. We also have Cyto-Select Plus, Cyto-Select, and Cyto-Select Mouse, which are the kit that you can pick to find the cells that express corresponding antibodies. We are also actively developing new kits in areas such as antibody discovery, cell line development, and cell therapy. And this QR code here, just you can take it and take you to the assay range page on our website if people like more information. All right, thank you very much for your time and attention. in this talk, I just want to briefly recap what I have talked about, right? So I have introduced to you the idea of pico droplet technology and why they are so good for isolating cells you want using Cyto-Mine platform, and explain how it addresses the need in rapid cell selection. I have also shown the data demonstrating our Cyto-Select fusion protein detection kit performance. So this webinar is just a sneak peek into our capability of supporting the fundamental need, which is to find the cells you want fast with ease. And there are a lot more seminars coming soon. So feel free to check out our website and contact us for more information. And we are happy to take any questions. And thank you very much for your attention again.
Okay. Thank you so much, Min, for that insightful presentation. We've got some really good questions actually from the audience, so let's dive into the Q&A. First question is, have you developed an assay for bispecific or multispecific antibodies?
Well, first of all, thank you very much for your attention. Yes, we are developing those assays. So the bispecific assay is being developed now, and it should be available, launched toward the end of this year, so Q4 for this year. As for antigen-specific assay, it's almost ready. So good news is that it should be launched in Q2 2024. So stay tuned.
And the second question, is FC Fusion Kit also applicable to the Cyto-Mine?
Yes, FC Fusion Kit is definitely available to be used on Cyto-Mine. Right? Because in that assay, which I show you, it's only required the thread analysis. So you definitely can use that assay on the first generation Cyto-Mine. But also you can also use on Chroma as well.
Another one. How many cells would I need to load into the Cyto-Mine?
That's a great question. So generally speaking, we tell people to load anywhere between 100,000 and up to 40 million cells per round. But it's really depend on what you want on your assay, right? So our technology is based on Poisson distribution, which is basically a statistical model used to estimate how many cells per droplet. So cell input is critical. So we recommend 3 screening modes: monoclonality mode, high throughput mode, as well as enrichment mode. So for monoclonality mode, you can load 100,000 up to 400,000 per experiment to maximize single cell encapsulation, which is great for cell line development, right? Where monoclonality is absolutely critical. Then for high throughput mode, you can load anywhere between 500,000 cells to 2 million cells. It is great for high throughput single cell screening. For example, for antibody discovery, right? Application which require a lot of cells to be screened. And then we also recommend we have a enrichment mode, which allow you to load up to 40 million cells and do a pool dispense, which you can tell the machine to do, right? So depend on your downstream application, you can do another run of a Cyto-Mine screening to screen the pool so that you can capture those rare cells for single cell dispense. So some of our customers, they are doing it this way to capture the really rare cell population from a huge, 40 million cell pool. Right? Hopefully that answers your question.
From the same person, actually, there's another one, and they've asked, how long can cells be incubated for in Cyto-Mine?
Yes, so you can, on Cyto-Mine, you can incubate for up to 24 hours. But it's really depend on the expression level of the molecule of interest, right? But generally speaking, for cell line development and antibody discovery, where the secret product is detected by thread, I mean, generally we see, 1 to 2 hours incubation is more than enough. But for assay, you might need a longer time. For example, reporter cell assay or internalization assay, you might extend it to overnight or up to 24 hours. But the good thing I want to emphasize here is that our software has this built-in capability where you can tell the machine to re-incubate. For example, say half hour, you see, oh, my expression level is not high enough, I don't see any signal. Yeah, you can tell the machine to extend the incubation to 2 hours or 4 hours, so on and so forth. So you can use this function to do a time point analysis on your stuff. So it's very easy to do. Right. Thank you very much. Next one.
You select cells in a very peculiar environment. How do you ensure that the clone you select as the best producer will also be the best producer in a different environment, e.g. a 2000-liter BR?
That's a fantastic question, right? So I would say there are a lot of factors that might affect the scalability here. So I think your question can be also the same as asking for scalability, right? For example, right, the viability, right? So our workflow and the reagent are very cell friendly and have minimal impact on cell viability. So among the users, I would say the outgrowth rate of a single cell disband is typically ranging from 30% to 80%, but it mainly depends on the cell performance and your growth media and many other factors that are out of Cyto-Mine, right? But we have done internal analysis where we compare the 130 experiments side by side using either manual dilution or using Cyto-Mine. What we have found is that the growth rate are identical to that of manual dilution cloning. So you don't have to worry about our Cyto-Mine process is going to kill your cells, right? That's not a question here. I would say another factor that might affect your scalability is whether or not the clone maintain the high expression level after being isolated, right? So on top of my mind, I know that one of our biopharma client has tried scale up to 50-liter bioreactor, which worked really great for them. So you can think about our pickle droplet is only 450 picoliter, and you scale up to 50 liter. Well, that's the magnitude of 12 order, right? That's a massive jump, and it still works in their hand. So that's why I'm thinking is that our machines really make it easy if you really want to jump it up to 2,000-liter bioreactor, because what we have been doing here is to find the cells that you want and really fast, because we can screen millions of cells just one day, and you can find those individual cells and you can test them, right? So that said, every single step is unique. So I would like to understand more about your specific goals and whether it's maximizing yield or ensuring cell viability or tackling a particular bottleneck at 2,000 liters. So if you can give us more information, I mean, we're more than happy to work with you to achieve your goal to up to a 2,000-liter bioreactor. Right? So feel free to reach out to us after this meeting.
Actually, on that note, if you have any further questions after this session, or if you'd like to follow up with Min on anything, you can reach out to us on contact@spherebio.com. So we have one more question before we go, and that is, are the pictures from the instrument sufficient to demonstrate the monoclonality for IND filing?
Yeah, it's definitely. So our software has this built-in capability of taking 5 continuous image for every single cell being disbanded, right? So to prove monoclonality. So that is built in with our software for every single cell is being disbanded. So it's definitely suitable for our R&D work. Not only that, but also we have this 21 CFR Part 11 built in our system, so that you don't have to worry about the electrical record, which is definitely required for R&D work as well.
Okay, I think that brings us to the end of our webinar today. Min, thank you so much for your time and your expertise. And thank you to our lovely audience for attending today and making the time to come and see us. There will be future sessions on future assays and other products, so keep an eye out for our webinars. Thank you. Have a good day or evening, wherever you are.
Thank you.